Oh, good question. Thank you for asking that. All right, here we go. I know it's not a cumulative final. It's just that last unit. So you don't, once you're done with this stuff, you're done with this stuff. I know that maybe that'll make your headache a little bit less than it should be. Okay, this is the banding pattern. It's a little confusing. Okay, and the stripes basically alternate what we call light and dark. What causes it, and this is what they look like. I'll get back to the slide. Is this the overlap? Here, the red ones are called thick filaments. We're going to get back to it and go over the anatomy of it. The blue ones are thin filaments. Where they overlap, because not much light can get through, it appears darker than here, where there's just a thin filament and it's lighter. So the fancy names, as you can see, are A bands and I bands. Can't ever get this wrong. Dark has an A, light has an I. You'll notice light doesn't have an A and dark doesn't have an I. That's all you gotta know. Do not get that question wrong. I'll be very aggravated. Dark bands, you know, there's no I in team, but there's an I in light. There's an A in dark. I used to counter that with my son who played goalie for about 6 years. In ice hockey, there's an I in goalie. I was in ice rinks every day of the week for like 7 years, early in the morning. My daughter figure skated. And she married a guy who actually was a professional hockey player. I'll tell you where he went to school? Robert Morris University. He graduated in 205, was drafted by the Pens in 7th round. Played at the minor league level, didn't make it to the big team. So I mean, he was very, very skilled. There's not a whole lot of players made the big team. My son skated with the guy who made the Penguins years ago. He's 43. He made it previously with the Penguins for a year from our area, our high school. He was on the high school team. So. And so her husband, he coaches hockey. He coaches some elite young, like teenage teams, stuff like that. So it's true. I know what I speak when it comes to the ice. So it goes around, comes around. Who go figure? She ends up marrying some guy from Robert Morris University. So the stripes, the A bands are dark. There's an H zone and M line. I'll show them to you. They're not important to me. It's kind of, and I'll show it to you. It's kind of where those thick fibers are kind of attached in the interior of the cell. And the I bands are lighter. And what you're going to see is what defines these little sort of boxcar units called sarcomeres are Z discs. And because the proteins zigzag, they were given that name. So, I mean, you can't really see it from here, but it's a very distinctive picture. This is the sarcomere. This is really the important part. This is how it works. Okay, it contains an A band, as you will see, and half of an I band, because one half is on one side, one half is on the other side of these Z discs, which are the boundaries. And the boxcar analogy is great. Because, I mean, if you, like I do, drive on 65 all the time, you get trains that are ridiculously long. That's kind of what this is like. And so imagine they're all shortening a little bit, a foot each one. Imagine how much shortening you get for the length of this magical length of that giant train. So this is what it looks like. The Z discs, this is a sarcomere. It's bounded by those Z discs. So they go from Z disc to Z disc. That light band made out of a protein called actin that we will get to, this thin filament in blue, effectively, the area where it does not overlap is equal on either side. And that makes it the I band. And so there's kind of like half an I band on one and half an I band on the other divided by the Z disc. The A band is where they overlap. The only place where it's not completely dark is in the middle, this so-called H zone where there's M proteins whose names you don't have to know. But that's kind of the middle area. And that has, and that's kind of like when you see it, when they begin to slide over one another, how far those thin filaments can really go. Take a look at this stuff. This is how densely packed. You'll see it a little better in another illustration. The red are thick, the blue are thin. So you see they sort of share. The reality is there's about a 2-to-1 ratio of thin to thick. That's why we can see it. When you get to smooth muscle, the ratio is like 12 or 13 to 1, thin to thick. So here you have a lot of thick fibers comparatively. Then you have like 1/6 when you get to smooth. They're there, you just can't see them. So it's a very orderly arrangement. Again, their names are actin and myosin. Actin, okay, like I said, across that I band and where they overlap into the A, that's what actually makes the A. They're anchored to the Z disc. The myosin are a little bit different, okay? And you're going to see the arrangement in a second. And the myosin, though they don't picture it here, the connection that red has is that magical protein that looks kind of like a coil or a spring. That's the titin, T-I-T-I-N. And I emphasize it because usually we spell titin, T-I-T-A-N. Okay, but this is T-I-T-I-N. And effectively, that's the extensibility portion. So a lot of times shape mirrors form and function. So here's what it looks like. Here's that titin. This is a great schematic. You're going to see the difference when it moves and when it doesn't. Here you can see that sort of coiled appearance, which is actually what it looks like from a micrographic standpoint. You can't really see it unless you use really sophisticated technology. Here you can see the actin, and it's considerably thinner. This is really multiple myosin strands. So the filament is multiple strands and appears much thicker than the actin, which is really, and the way I always use the same description, it's like a string of pearls that you've twisted. It's almost like a double string of pearls, that you would say. If you can imagine, I don't know how much people wear pearls today, but my mom's day and age, that was a big deal. My wife, and I think she's not a jewelry person. Saved me a lot of money. So here, the I band, why it's light, you just have thin filaments. And here you can see the A band, you can see that overlap pattern more clearly here. And this is in the middle. And again, because you don't have fibers overlapping, it's relatively clear, this midline area. And there's lots of different terms for it. And I don't really want to go crazy about it. So the myofilaments, the myosin has, and when we talk about the so-called thick filaments, you have different kinds. They're all made of polypeptide chains. For right now, it's not a big deal about heavy and light. So effectively, the way they intertwine creates the heavier portion of it, the tail, and then you're going to see the heads. And what you're going to see effectively like this, the myosin, I'll flip and show it to you individually. Oh, I'm going backwards, I'm sorry. That's what it individually looks like. It's twisted, we braid things for strength. That's the heavier chains. You don't have to know that they're heavy or light. And the hinge region, the heads are on there. And those heads are what effectively are going to move. When they're at rest, they're like this. All muscles are ready made to fire. In order for a muscle to relax, we reload it. We're going to go over this at length with energy, so it's ready to fire again. And so it's like this, the way that you can see it in this illustration, it is cocked, is the expression we use. When it is cocked like that, it's preloaded with energy, and all it has to do is basically, as you can see on top of it, these are called binding sites. We name things not only for what they're on, but what they're going to attach to. The two parts, the thin filaments are called actin, the thick ones are myosin. So we have binding sites. The best analogy is like when you study the bones. You had the radial head. What does it articulate with? The radial notch, which is on the ulna. So a lot of times we'll name something for what it's going to attach to. The radial notch on the ulna joins the radial head on the radius. Same deal here. you have, these are called actin binding sites. And you're going to see when you get to actin, they have myosin binding sites. They're almost like this next to each other, that kind of close proximity. And you'll see that once we remove a particular protein that's covering those binding sites, they immediately bond together, and immediately that does this. It takes that head that we call cocked or ready or activated and does this. And effectively, it's because they're attached to those Z discs. It basically, those heads pull on the red, cock and go this way toward the midline when they're attached to the blue thin filament and pull it toward the midline. So that's how it shortens. It's really how muscles work. We are going to see a series of animations kind of show you how this works when we get to it. And that's really most of what we're doing today, is to go through that. So again, this is the sarcomere. You saw that illustration before. The actin are thin, the myosin are thick. So the thick filaments like this, and what I was talking about is called cross bridging. You're going to see our studies, and this is, frankly, this is all introductory. I mean, as much as it seems like I'm spending an enormous amount of time on it, and I am, it's introductory. What you're going to see with this, everyone, is three processes. We're going to see what's called the neuromuscular junction. We're going to see how the nerve tells the muscle to contract. A multiple series of steps. I'm just giving you the terminology and kind of a basic understanding of this magical thing we call sarcomere that actually is where it slides and the movement is created. Number two, we're going to see, and this is the animation we're going to look at today, called excitation, contraction, coupling. I'm showing it to you to illustrate a couple of things. I'll go back and really show you what it means next time we get together. That is, and these animations are excellent in this particular PowerPoint sequence. And then lastly, that's going to be the translation of chemical energy into mechanical energy. Taking the energy we get from ATP and magically making the muscle shorten, the sarcomere shorten, and creating movement. Lastly, we're going to look closely at the movement where it forms the cross bridge, called the cross bridge cycle, which also involves energy to release them and to recock the head. A lot of energy in muscles. That's why there's so much oxygen, why there's those glycosomes, why we have a lot of waste products. Because you have energy, the nerve has to have energy to signal the muscle. The muscle, as you will see, is going to be, you're going to see that energy is going to translate and release calcium that we're going to look at today, that's stored inside part of the muscle, part of the sarcomere, and release it, and that is going to cause it to fire. And then we're going to need more energy to make it relax, and we're going to have more energy to pump the calcium back in to where it's being stored. A lot of energy. It goes into muscles. And the whole idea of this offset, just to complete the thought, and you're going to see it better in not this animation, but others, about how it's basically like a phenomenon, like a ratcheting phenomenon. Muscles, if you have to lift something light, very easy. You don't have to use a lot of muscle power. But if you try to lift something heavy, like a desk or even a chair, you have to basically use more and more power. And to do that, you have to shorten and shorten the muscle to almost to its maximum. So it's a little bit different. So all this together, the actins we talked about in the cell, making microvilli, pseudopods for exoendocytosis, amoeboid motion, all of those are the same, except in that animation I showed you, they can form and unform. These are formed, they're not going away. They're made of, the individual ones are called globular actin, and they form a strand we call filamentous or fibrous actin. The F would go for either one of those words. And those actin strands then twist. This is like the string of pearls that looks like two. And what holds them, and this is what we're going to get to, not to go touch it today and talk more about it next time, particularly with animations, is tropomyosin, which is called a regulatory protein, and troponin that holds it in place. I'll endeavor to show you in a second. We'll come back to those names, do the animation, and try to summarize this in a long-winded way. So here, again, the heads are not all in the same place. So when one detaches, this one might stay attached, and then the next one will attach, and so on. So it allows, it's almost like taking a rope and it's like if you're trying to climb a rope, you always keep one hand on it and one hand off. If you took them both off, you're going back to the bottom. Biologically, it's kind of the same thing. That's why they're offset. So here is really where, even though the myosin is what moves, is the motor protein, this is how it moves. That strand, notice those are the binding sites. In this strand-like protein or filamentous protein called actin, the active sites are covered. Once they're uncovered, they're so close to the ones on myosin it fires. This strand-like protein is the way I don't have anything better to show you than this. Covers, it's like as though, the cover on my phone, along the edges is where those active sites are. The strand's there. Troponin, tropomyosin is a strand-like protein. Troponin are little plugs, toothpicks, thumbtacks, push pins, holding it in place. You're going to see when calcium floods the area, it alters or changes the troponin. Anything that comes in contact with a protein and bonds to it alters it, and it basically changes its shape. And what it does, rather than holding it there they go. So, I mean, you wrap something around it, if it don't secure it with something, staple it, tack it, toothpick it, it doesn't matter. It's going to come away. The whole idea of controlling this motion is you don't expose those heads until you're ready to use the muscle, biologically speaking, to try to give you an idea of how this whole assembly system works. So there's the titin. That holds the thick filaments. That's all about excessive stretch, recoil. This is this protein. You don't have to know these names except this one. There's other proteins. This dystrophin. Dys means something that's wrong or irregular. If you don't have the ability to do something, you have a disability. So in the body, dys means something is not right or not working properly. Trophy is growth. So atrophy without growth is something that happens when muscles are really damaged. They stop being, they actually shrink or shrivel. So dystrophin was named for the disease, muscular dystrophy. It's the protein that's affected. So dystrophy in this case means an inability for the muscle to grow properly. It begins to deteriorate early on. And unfortunately, eventually the muscles that get involved as it ascends are respiratory and heart muscles. And that's why people with this disease succumb, they die at an early age. This is, I pronounce it Duchenne. Some people say Duquesne, don't know why. It's the most common and serious form. It destroys the muscle, and the muscles deteriorate. They're replaced by scar tissue and fat. It may look like somebody has muscles, but the muscles are effectively not muscles whatsoever. It is called sex-linked recessive. That means it travels, if you remember your basic genetics, on what's called the X chromosome. Women have two X's. And so the X that has the disease is masked by the other X. We never see two X's together because, unfortunately, that would require two individuals that both have the disease, and it's very unlikely they would be able to conceive children. But men have a Y and an X, and the Y doesn't have anything to mask it. So 1 out of 3,600 is by no means common, but it's common enough that it can rear its ugly head. So it's something that you may or may not see. It's one of those diseases we don't understand really well. It's probably genetically based, as it is, but probably it is genetically based. It starts mostly in boys, again, nearly exclusively, and they start to grow normal, but this dystrophin begins to accumulate and damage the muscles, as you can see, early, and then eventually chest, respiration, pneumonia, heart problems, all. Years ago, the 30 to 40 was sort of the end range. We now see it moving greater. I told all the classes this. We do this in micro. Years ago in the 80s, about 1988, they came out with a piece of legislation that was brought about by AIDS. You see, AIDS, basically, pharmaceutical companies don't do things for fun. They do things for profit. So Big Pharma had no incentive to make medications to treat HIV. A very limited amount of people had them, much less than people think. There were a lot of celebrities who had it, who, by our parlance today, would have been called closeted gays, mostly, because that's who it affected most at that point. And what happened, because that was an important community, and these folks have Hollywood entertainment, have a lot of money, and they have a lot of prestige, and they know the right people, and they donate to the right campaigns. They got Congress to pass what's called the Orphan Drug Act. It has nothing to do with orphans. It is basically an incentive for big pharmaceutical companies to fund research, and it will fast track medications, particularly then for HIV. But it applies to a lot of other things. ALS, which is Lou Gehrig's syndrome, amyotrophic lateral sclerosis. For you Grey's Anatomy fans, my wife tells me that's affected Dr. McSteamy as opposed to Dr. McDreamy. She just ignore her. She's in touch with her inner cougar. Trust me. I'm sorry. Her major crush is Tom Brady. I'm sorry. I know I look close. And so what happens is this ascends, it becomes a problem. So they live longer now. And in micro, we go over the impact and kind of I'll give you the sort of the history lesson about HIV/AIDS, because age-wise and where I was located, I have a particularly large knowledge subset about HIV/AIDS. Moving on. So there's they named the gene dystrophin because of what it did. And you don't have to really go into what it does in the sarcolemma and inflammation and blah, 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 blah in the picture. Okay. What's interesting, and this is the confusing part, and we're getting near the end of this discussion. What happens, remember, it's called the sarcomere. The membrane is a sarcolemma. The sarcoplasmic reticulum, it doesn't really have rough, it has smooth. The smooth endoplasmic reticulum is filled with calcium. The calcium, when it's released, is what triggers the troponin to change, allows, and I won't do it again with a cell phone and a wire, the cord, to slide off and expose those heads. Once they're exposed, they come together immediately, and we have what's called sliding filament theory. The fact that they're at the A and I band is not a major factor either. What they're associated with, and the animation shows it very, very well, are what are called T or transverse tubules. They're infolds of the sarcolemma. The sarcolemma is the cell membrane. Okay, and what that is it's a way of, remember, it's all about conducting an electrical signal. This is how the electrical signal gets conducted. It gets conducted not only along the longitudinal or the long axis of this fiber, but it has a way of going inside. So it's folded in and it surrounds and envelops all of the fibrils. You will see that. And what they form is a triad, and you're going to see that. And what it is basically, it's easier to show it to you than go back to this. So let me show you. I'm going backwards, not forwards. So it's kind of what it looks like. This is a triad, where this in gray is the infolding of the sarcolemma rather, basically the cell membrane. And then in blue, this is the sarcoplasmic reticulum. These sort of bulges here and here are called cisterns. Cistern is an old term for a well. So they're called terminal cisterns or terminal cisternae, and they form what we call a triad because you have three structures, one on each side, a cistern in the middle, in the middle, the transverse or T-tubule. Transverse because it's going at right angles to the long axis of the cell itself. You're going to see that in the animation. So let's go over to that. And then we'll finish up with that stuff. So we're getting close to the end of this marathon. I just have to go through all this stuff because this is the stuff that's coming, and it's unbelievably complex. I mean, just here, just alone, just to give you an idea, we go over all this myriad of steps about how a nerve fires. All that stuff that you're seeing here. So there's a lot in this. Oh, I missed one. Sorry. This is it. Let's see if we can hear it, hopefully. Not all just the sound. Bear with me.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
So to me, they're really good animations. And just to give you the highlight of what, and we'll see this again, because then you'll be more aware of actually what's going on and how this thing we call an action potential occurs. Its name tells you everything. It's about a potential for action. We reach enough. It's like, I guess maybe the way it's making a decision. I'm thirsty. I'm not that thirsty. And maybe whether I'm going to go up and get a drink or I'm going to just grab my drink and have a sip, I don't know. So the action potential is where I'm actually deciding I'm going to grab it and drink it. And everything else is sort of, and then we have, it's almost like you're deciding. So you have potentials that are not quite up to producing an action. But once an action potential occurs, it goes right through to the very end. So that's sort of a little, just an idea. And that action potential in the case of this is that waveform of electrical activity first going through the nerve and then going through the muscle. As it goes through the muscle, it keeps rapidly changing the membrane from positive to negative, flipping it negative to positive, just like you're flipping a switch. And that switch, that electrical current, eventually, as you saw, will trigger something that's electrically, or what we call a voltage gated channel or a voltage sensor channel, to create an opening and allow that calcium to flood. And voila, once the calciums are there, the troponin alters its shape, the tropomyosin slides out of the way, and then so we understand it. It's just trying to get you to have an understanding of it. And I realize it's complicated. Those are those infolds. These are transverse. The long axis, as you can see, goes along the long line, but the infoldings, the way that it's passed as the signal goes through on the outside, it surrounds inside. Those are all the fibrils, all those bundles of those flexible rods inside. And so that's real, and it's a really good illustration of it. As we move it forward, now you see the sarcoplasmic reticulum. It's filled. all of these areas are filled with calcium. The calcium in a skeletal muscle is inside. We call it intracellular. When you talk about what makes the hard work or smooth muscle, it's calcium from the outside. What makes a nerve fire? Calcium on the outside. So you're going to see now very clearly the role of calcium, why calcium levels, and this is the triad. It's a juncture point, and you can see it when the signal goes through like that and goes down. Basically, you can see the bulges there. Those are enlargements. They call it cisterns or wells where calcium is stored. And then you have the T-tubule, and you could just see that signal go by. When the signal goes by, graphically, it'll show you, bang, it opens this protein. Proteins change not just by chemical or by pressure, but this is electrical. Hence the name voltage sensitive or voltage gated channel is what you're really going to see. And then it allows the calcium to go out. Eventually you have to pump that calcium back in, expend energy to basically make the muscle stop contracting. You can see that's the contraction. So
Ions are responsible.
Yeah, we heard you, lady. Take a rest. I turn the sound down on commercials. I'm tired of them. This is one I laugh at. This is what I mean about all these steps. This is next time. I know. You can be like my middle child, who's a wonderful guy, but when it comes to school, he goes, all that stuff makes my head hurt. So here we are again, just about back. So, voila, almost there. So the whole idea of all of this is called the sliding filament model. They slide over one another. And so as they don't shorten per se, the sarcomere shortens. The actin and myosin stay the same. So neither thick or thin filaments change length, but when these cross bridges form, they slide. In the process, the contraction occurs of the sarcomere. So they form and break several times. That's what I mean by way they're sort of asymmetrically placed. They're offset. So while one attaches, the other one detaches, another one is attached. So when you have to use that muscle more, well, I was trying to lift that. I thought it was light, but it's really heavier. And now that's really what happens. And we bring it to its endpoint. So what you're going to see, the boundaries of the Z disc are pulled closer to the middle, like the walls coming in this way or that way. Visibly, the I bands will shorten. And it'll look like the Z discs are coming closer together. I'm not too worried about it. H zones. And the A band, I don't know if it moves closer. It just looks different. So here it is at rest. Completely at rest. Actual micrograph of this. Here you can see the dark zone, that sort of mid or H zone, kind of look like a little letter H in a funny way. And you can see the I band, how light it is compared to how dark it is, the A band. And you can see there's about half of it is on one side, and the other half, if they had it expanded, would be on the other side. The physical limit of how much this can shorten, these Z discs can move toward the middle, is what this gets here. The actin filaments get here and here. These aren't going to change, but when they relax, that tighten over here, as you can see, that will courteously, if it's stretched, it'll bring it back to the resting position. So again, typically when we relax a muscle, we don't have to worry about it. Okay, we let these things just go back to what we call its resting state. So I flip-flop this quite a little bit. Here's the Z disc. Look what happens when I advance it. Boom. Back again. Here's the Z disc. Look at how much white is there now. Look how much white is there now. Very, very little. So the boundaries are here and here. Look how far the boundary moves when I go backwards. Now you can see quite a bit of white. When I go forwards, very little. At the same time, even though there's a color change somewhat in this fully contracted, but fully contracted means just this. The actins have been pulled toward the midline because of the proteins, they can't go any farther. This is maximum overlap. Yeah, that midline it doesn't have that clear area, but not a big deal. But this is really where the motion is occurring. It's bringing those Z discs closer together rather than a resting state farther apart. So effectively, it's shortening these sarcomeres. That's the so-called sliding filament theory, and it's there. It's how it works. There's no debate. It's still called the theory. So how do we make this all occur? We decide. That's what voluntary is. I'm going to reach for the mouse. I'm going to reach for my phone. I just got a message. I'm going to tell you I got to call. Hit the old, can I call you later? Whatever. We've all done that. So the signal, and it's a two-nerve circuit. It goes from your brain, usually to your spinal cord. Let's say we're talking about extremities. And basically from the spinal cord out to wherever it is. So I'm going to move my arm. It's from roughly this area of my spinal cord out somewhere in my arm or foot. From my low back, it would go into my thigh, my leg, my foot. But we voluntarily control it. We make that decision. Once we make that decision, that nerve, the first nerve in the circuit going to the proper area of the spinal cord, fires, and that electrical signal moves. In order to tell the next nerve, it has to, it's not like an electrical circuit. Oh, I just plug in and it goes on. It doesn't work that way. It's not like you have a power strip. You plug something into it. That's electric to electric. Nearly all of the time, your nervous system releases this. It releases something called a neurotransmitter. You're going to get sick and tired hearing about neurotransmitters. Thankfully, the single most important one to know about is this one. This is the most abundant. This is the one we understand the most. This is all your skeletal muscles are triggered by this. A lot of your smooth muscles are triggered by this. It's in your brain as well. It's everywhere. The reason your heart is under control and is racing, your vagus nerve. You probably remember that from lab. The 10th cranial nerve. What does it release? It releases acetylcholine. That slows your heart rate down. That's how it works. We're going to look at that in autonomic nervous system at the very end of the class. Got it? And acetylcholine is a very interesting chemical. It's made from what's called the acetyl group, which is acetic acid is vinegar. It's not like we have tons of vinegar flowing through our veins, but the reality is we have a breakdown product called acetyl, and we use choline in a lot of places. So it comes together and forms this, and it's the only one in its category unto itself. So neurons and muscles are excitable, and they can reach these what we call action potentials. That's that membrane, sort of, and conduct the impulse along the membrane or inside. And then the acetylcholine is the key, sending the message at the neuromuscular junction where the nerve basically tells the muscle, fire. At that point, acetylcholine is released. How much is released, how rapidly it's released, determines whether it's thinking about getting a drink versus going up and getting a drink. And we'll get to that as well. Because we have something that don't even, just ignore this next phrase, end plate potentials versus action potentials. It'll only confuse you more. I'm sorry. So the big deal, and you saw it, okay, the acetylcholine is a chemical. So what it does to make that muscle fire, it alters a channel that allows charged particles to move. That's called a chemically gated, and you will often see the term ligand gated channel. The terms are interchangeable. Acetylcholine is the ligand, the chemical in question. And you also saw, okay, voltage gated channels right now. Go back and look at that animation, and we'll look at it again. It went by that flip-flopping of the membrane, triggered that channel to change shape, and the calcium came out. That's a voltage sensitive or voltage gated channel. Voila. Here would be the acetylcholine opening that up. And you're going to see it's two-way traffic involving sodium and potassium. We get there next week. Or in this case, this is opening that passageway, let the calcium do its thing. With this little ball, when we think of gated channels, your gate doors open and close. That little ball, particularly important in the heart, is called an inactivation gate. Don't know about that yet. It's like locking the gate. So like at night when we finally get the cat either out or in, we do the back door. Cats. But he's a cat. The other cat doesn't go out as much. We often see pictures of the cat on TV. We think he probably has an acting gig or a photo op going on. He's connected. So we're going to look at all of those things. We're going to look at the anatomy of it. This is what's coming up. So brace yourselves for next week's presentation. We're going to see that in order for a muscle not to fatigue, if we kept triggering the same muscle in the same place, so what do? We trigger it in different areas, different fibers, different cells, which prevents fatigue. We'll go over that. We're going to look at how the chemicals are released. We're going to look at the makeup of how the chemicals come across, what triggers them, how they start. All these little gateways here will be explained. And we're going to look at, again, we'll probably get to the events at the neuromuscular junction. And for you, since we do a double class, we'll probably see that excitation, contraction, coupling. I don't know that we'll get to the cross bridge side. There is a lot here. So I wanted to let you know about how extreme this is. Look at all the steps that are involved. And so when I explained to the other class, what you see, I don't know if you had them in anatomy in the lab. We have what are called sequence questions. It's like the sequence of events. Neurotransmitters released, there's an action potential, the muscle contracts. That's the sequence of events. In lab, like an AP2, typical question, okay, where's it, a drop of blood from your heart, how does it get to your big toe? Okay, it goes into the aorta goes into whatever blood vessel, goes into the common iliac artery, and then it goes into, etc., the external iliac and the femoral and blah, 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 all that. That's kind of how a sequence question, or you trace a drop of blood from when it starts going all the way back to the heart. So you look at the sequence of events or how urine is produced. This is the kind of question, and I won't go into all the details, but I'm going to do major events, and students have always found that to be complicated, difficult to remember. So that's why we go over all this stuff. And because you see this repeat pattern all the time, you're going to get sick and tired wherever you're going, particularly if you're going in healthcare. If you know anybody who has obsessive compulsive disorder, eating disorder, mild depression, they're all taking major depression. They're all taking medicines that affect neurotransmitters. All of them. Doesn't matter. Same thing. So if you hear anybody who's on, in the old days, it started with something called Prozac, and then it went to Paxil and Zoloft, and they're probably still used. All of those affect neurotransmitters. Usually they make more of them available. And we'll get into why they work. And then I reinforce that when we do the neurology situation. This kind of subject matter I'm pretty familiar with. Muscles and nerves, I have a lot of information about to share with you, I hope. So we will conclude it there. My voice is given out. It's only 4:45. And I will send you what I present on Friday to the other classes. So they will probably get some more of this. So I'm sure on Friday, I'll probably start to look at that next animation and starting to go over all of these steps at the neuromuscular junction. Okay, so again, the test is next week. Any questions that you have, shoot me an email, whatever you want to do. Just give me a holler, please. And we'll go from there. So again, we do the test first and we do the rest afterwards. Everybody's having the test on the Wednesday. Thank you.